you a basic understanding of how the biological fungi trichoderma interacts with crops. And so even though we may be talking about corn or soybeans, basically trichoderma has the same interaction with any type of crops. So these first few slides are important to be able to understand how trichoderma interacts with crops. So first we're going to talk about Sabrex for corn and Graphex SA for soybeans. And both these products contains the trichoderma that I was talking about. So basically what trichoderma is, it's a microbial, and it's found in soils all around the world. But we found that not all trichoderma strains are beneficial to crops or to plants. And so based on more than 3 decades of research at Cornell University and working with Dr. Gary Harmon, and by the way, Dr. Gary Harmon is retired from Cornell, and now he's our Chief Scientific Officer for ABM. What they have done over the past decades here have found technology that now they can identify and isolate specific strains of trichoderma. And it's kind of like a seed company that would identify and isolate an inbred for corn, okay? And so then what we do, once we've identified these different strains, then we can take the different strains and test them with various crops, or take combinations of strains and test them with various crops. And so that's what we've been doing at ABM, and now we've been able to come up with some products that are beneficial to crops out in the marketplace. Also ABM, you know, in the years past, has taken a long time to identify and isolate these strains and find out which which genes, microorganisms are beneficial. So ABM is also working on new technology where they can screen thousands of strains of microbials and analyze their genes and phenotypes to be able to identify which ones would be beneficial crops without spending lots of money and a lot of time treating seed and put it in and doing field trials. So based on this new technology that they have, we can screen them and put them right into products and minimize the time that it takes to bring it to the market. So how does trichoderma work? So basically what trichoderma is, it's a spore former, and a spore, when applied to seed, it'll actually grow with a plant root system all season long. So it'll keep reproducing itself in the root system. So what they do is they colonize with the crop root system. And what they'll do is they'll go in and they'll start feeding on the plant roots, and they're going after the starches and sugars that the plant produces. And at the same time, they'll exude enzymes and proteins that the plant likes. And as a result of that, we find we get a larger, more fibrous root system. And as a result of that larger, more fibrous root system, we find the plant then is better able to take up moisture out of the soil. It's better able to take up nutrients out of the soil. The exudate that I was telling you about actually helps put a biobarrier around the root system to help protect the plant against diseases like Pythium, Rhizoctonium, and Fusarium. So as a result, we find we get healthier plants. We find that we get improved germination, improved emergence, and seedling vigor with the plant. As a result of that colonization, we also find that we can will affect the physiology of the whole plant, even the foliar and then leaf biology. And I'll show you some pictures of that examples here in just a moment. On this next slide, let's look down here at picture B. And right here you can see where that spore is, and the trichoderma is growing out of that spore, okay? And then the center seed, G, or center slide G, you can see where the trichoderma is actually boring itself down into a fine root hair, okay? And then over on the right, you can see a cross-section of the root where we've cut the root in half. And you can see here where the trichoderma is boring into the root. And here the trichoderma is inside the root. And it's a zone right around the trichoderma, okay? It's where all the biological transfer of information happens between trichoderma and the plant. This graph here is got a lot of big words on it. I'm sure a PhD designed this graph, but basically what we want, the point we want to make out here is that trichoderma is only found in the root system. Trichoderma will never go up into the plant. those microorganisms will always stay down here in the root system. Now, that biological transfer of information then can be systemic, and the plant can take out that information in it, and that's where we start seeing physiological changes in the plant. Well, as a result of that colonization, we find we get priming defense from diseases. We get increased photosynthesis and carbohydrate metabolism. We get abiotic stress response in instances when we get into a drouthier situation, drouthier fields. We also find we get improved plant nitrogen use efficiency. The plant becomes more efficient in taking up the nitrogen. The first product I'll talk briefly about here would be the Sabrex for corn. And the Sabrex for corn, well, here it shows our research and development selection of the various strains of trichoderma and how we combine them together and find out which one of these is working best for, in this case, a corn plant. Here again, we talk about physiological changes. Same hybrid planted the same day, side by side. This side over here on the right-hand side was treated with Sabrex, and already at this early growth stage, we can see a larger, more fibrous root system. And actually, we're probably about a whole leaf stage ahead of the plant on the left-hand side. Then we look at stalk health. We can see the differences between the stalk between the two. Here again, there's two plants on each side, but again, we can see a physiological change in the plant where we get larger, more fibrous root system. When we say we get the trichoderma, when we see physiological changes in the plant, it's kind of like what we're doing is that trichoderma is helping the plant express the genes that it already has better. And so we're just bringing the genes out that the plant already has. In this photograph here on the right-hand side, you can see it was treated with Sabrex. On the left-hand side, it was untreated, same hybrid. At this early growth stage right here, about the 2-leaf stage, we can already see the plants on the right-hand side tapping into the nutrients and the nitrogen, because you can see that the side on the right is a greener corn than what's on the left-hand side. Effects on seedling germination. Now this is a field of pickles, but these two rows here were treated with Sabrex, which contains trichoderma, and the other rows over here on the left were untreated. So we find that we get increased germination and better early season vigor with the plants. Changes in plant physiology. When we take a cross section of corn, the left side we can see here we've got much healthier stalks. We get healthier stalks. We're going to see better standability. My co-part out in Minnesota a couple of years ago, he said he went to look at a complaint where a field of corn was down and the guy had split his planter with Sabrex, and as we got out there, they hear me? Okay, can you hear me now? All right. He told me that the corn that was treated with Sabrex was standing like a fence post, and the untreated corn was all laying down. So we find we get healthier stalks, we get better standability. Just to give you some data information, this data here was collected in Indiana, Ohio, and Michigan. Those are the 3 states that I cover, and this is on 4 years of data. 51 locations. We're finding a 10.6 bushel trial advantage over non-Sabrex treated corn. Both corns, same hybrid, had the same base treatment from the seed company. Only difference was this side was treated with Sabrex and the control was not treated with Sabrex. As a result of that 10.6 bushel yield advantage, we find that the net profit to the grower then would be $38.67 per acre, and that's figuring on $4.20 corn. When we take those 51 locations and put them on a chart like this, we'll find that we don't always get a yield response. But we can see here that approximately 80–85% of the time, we do see a positive yield response with Sabrex for corn. And in summary then, return on investment, you're going to find that you're going to have fewer replants out there in the field, because we actually can improve the germination and emergence of the plants. A typical return on investment to the grower would be 6 to 1, or about $30 an acre to the grower. Now those numbers will change based on the commodity prices, and these numbers were set a couple of years ago. The Sabrex for corn comes in 3 different formulations, and the formulation that everybody out there is going to be looking at would be the planter box. So it would be Sabrex PB, and what that is, it's a talc graphite formulation as the carrier. And the grower's only going to be adding about 1/2 ounce per seed unit, and 1 pouch will treat 24 units of corn. And there are 6 pouches in a case, so a case is going to treat 144 units of corn. Each case will have 6 measuring cups in there, so that the grower can actually measure the product out and mix it in. It's important that the grower mixes the Sabrex PB in with his corn, so that he gets the full benefit of using Sabrex. We all know that farmers, when they're using talc and graphite, they'll fill the boxes up and then they'll come back and top it off with talc or graphite and close the lids and go. Well, that may be fine, and they get along okay with that, but with Sabrex, they really need to take a few extra seconds at each box and mix the product in well. I get a question asked a lot, does the Sabrex replace the talc and graphite that I'm already using? And my response is that keep in mind that our application rate is only 1/2 ounce per acre, whereas depending on their planter, they may be applying, the recommendations may be up to 3 or 4 ounces per 80,000 kernels. So it's probably not going to replace what they're doing, but it's going to complement what they're already doing. These other 2 formulations are for commercially applied by the seed company, so that wouldn't pertain to anybody here. But keep in mind that the Sabrex for corn has a 2-year shelf life in the container as well as on the seed. Okay, now we'll move on to soybeans, and the product we at ABM offers is GraphX SA. And so GraphX SA contains the non-public strains of Brady rhizobium. They were developed by a guy by the name of Dr. David Kirkendall, who spent 35 years at the USDA, and his total focus was on Brady rhizobium botanicum. And so out of his research and testing, he developed some strains that are proprietary only to ABM, and that's what we're using in our products today. We feel that these strains are like super strains and that they're more suited for the genetics of the soybean in today's market. We call it a triple stack inoculant because in GraphX SA we've added 3 different Brady rhizobium strains. And one of those strains likes the hot, dry soil conditions. Another strain likes cool, wet soils. And another strain likes nitrate carryover. And so by doing this, we help the grower spread his environmental risk out when he goes to the field and plant, because we know every year is going to be different. The other new thing that ABM applied for a patent on one of those strains, and we were awarded a patent this last year that states that one of those strains is actually glyphosate tolerant. When we look at nodules on a soybean plant, what you see right here would be typical of what you might see with resident rhizobiums that may be already in the soils or our other competitors' types of rhizobium. A lot of our competitors will go to the USDA bank and just pull out Brady rhizobium that's actually open to other industries, but these are not the strains that ABM uses. The strains that we use, you'll see here, are quite a big difference. Our strains are going to be larger in appearance. They're going to look like a crinkled up paper, rough look, which also allows for a larger surface area. You're going to find that the nodules on our inoculants will primarily be on the tap roots of the plants. So we find that these, by being on a tap root, the nodules are actually more efficient to the soybean plant. So again, they're larger in size, so it's a bigger factory. They're a wrinkled paper appearance, which allows for more surface area for converting atmospheric nitrogen into a nitrogen fusible to the soybean. You're going to see fewer nods per plant, so there's less mouths to feed. They're more efficient than older genetics of Brady rhizobium, and these genetics are very aggressive. And some trials we've done here in Indiana. This guy here used Graphex SA and the regular Graphex and the control. And between the Graphex SA and the control, we found a $50.45 return to the grower on his investment. So we found a 4.29 bushel yield increase. And we figured this number based on $12.60 soybeans. This trial here was conducted by Ohio State University in their inoculant trials. They tested this in 6 different counties, replicated in each county using the Graphex SA and a control. And as a result of all that average, we averaged out 3.65 bushel per acre advantage, or to the grower would be a net per acre of $40.92. But across the ABM, across the U.S., our 4-year average, we're finding a 4.51 bushel yield advantage. The Graphex SA contains those 3 Brady rhizobium strains, plus the SA means we've also added the fungi trichoderma. So it interacts with the soybean plant. This is the same as what it was when I was talking about corn. So actually, this product here is a 4-way. You get 3 Brady rhizobium and 1 trichoderma in the packet. It comes in a pouch like this. The application rate begins at ½ an ounce per 140,000 seed unit. 1 pouch will treat 50 units of seed, and there's 6 pouches in a case. A case will treat 300 units of seed. Now, on this product, we can only get a 1-year shelf life in the pouch, in the container. And the reason being is that the Brady rhizobium is the weakest link in there. If it was just trichoderma, we could get 2 years. But what we find is that Brady rhizobium is a weaker microbial, and we can only get a 1-year shelf life on it. So in this case, you got to use that or you're going to lose it. Each pouch has an expiration date. Anytime you're using any type of biologicals products, you need to look for an expiration date and make sure you're using that product within its before it expires. After that, you just need to pitch it, throw it away, because it's not going to be any good. We'll move on now to wheat and cereals. We have a product called Sabrex. We have a product called Sabrex for wheat and cereals, and so it can be used on wheat, barley, rye, sorghum, oats, spelt, triticale, and basically any type of small grain grass type crop that you may be growing. And again, this Sabrex for wheat, it contains 2 different types of strains of trichoderma, just same as the Sabrex for corn. It also has 2 types of strains of trichoderma. However, the strains in both products are different. Plus, the Sabrex for wheat is a different formulation because we're dealing with small grains, so the application rate is going to be different as well. So you can't mix the 2 back and forth with crops. So Sabrex for wheat and cereals needs to be only used on small grains. And again, as a result of that trichoderma, we find we get a larger, bigger root system. We'll get increased tillering. And from things that I've seen, we'll see about 10% tillering, more tillering. And so as a larger, more fibrous root system, we also help increase the soil organic matter and help in reducing soil erosion. And again, I'll show you some pictures here in a minute, but as a result of that trichoderma, we can find we'll get better germination, and the plant becomes more efficient in its nitrogen uptake. But typically, we'll see about a 4 to 5 bushel yield response on wheat. So benefits if it's a cover crop, though, I mean, if you're planting wheat and any kind of grass type crop as a cover crop, you're planting it because you're trying to scavenge nitrogen and you're trying to take up the nitrogen. So you're going to, like when I was telling you earlier, the trichoderma makes for a larger, more fibrous root system, and it also makes the plant more efficient in its nitrogen uptake. And it also helps reduce nitrogen leaching. So basically, by using Sabrex for wheat and cereals, either as a crop or as a cover crop, it just amplifies what a grower's already trying to do. And again, larger, more fibrous root mass, that helps to reduce soil erosion and builds organic matter, which again just amplifies what the grower's already trying to do. In this picture here, the outside 30 feet was untreated, and the inside of this field was treated with Sabrex for wheat and cereals. And you can see here in the front of the picture, the first 30 feet, we've got scattered emergence, okay? We've got a thin stand. Even back here by the tree line, you can see a thin patch coming down along that tree line. But then everything here in the middle was treated with Sabrex, and it's all nice, lush, and green. So another example of better seedling germination and emergence, and the plant's already taking up nitrogen. We talk about root mass. Here's a photo on the right-hand side where it was treated with Sabrex, so we get a larger, more fibrous root system under the plant. Here we can see an example of a more efficient nitrogen uptake. The rows over on the right-hand side were treated with Sabrex. On the left was untreated. On the left side, you can see a light green color to the wheat, whereas on the right-hand side, we've got a dark green color. So already at this early growth stage, the plant's already tapping into the nitrogen that's there in the soil. And as a result of this, in this case, we found a 6-bushel yield advantage. Here's another guy from Clinton County, Indiana. Same variety of wheat in the same field, treated on the right, untreated on the left. Just had a big difference in that field. On average, with ABN, we're seeing a 5.3 bushel yield advantage across the U.S. And it comes in different formulations again. So what most of you out there would be getting for your customers would be the planter box treatment. It's a talc graphite formulation. All of our formulations in talc graphite are 80% talc, 20% graphite. And the application rate here is 2 ounces per 100 pounds of wheat or cereals. And so a case contains 6 48-ounce pails, and a case then will treat 288 50-pound units of seed. These other two formulations, AC and LQ, are high concentrated formulations that a commercial treater would use to apply to the wheat when he's applying other insecticides and fungicides through a treater. Again, the shelf life on Sabrex is a 2-year shelf life in the container as well as on the seed, because it's all just straight trichoderma in this. We'll touch a little bit here on alfalfa and clover. We've got a product called Graphex SA for alfalfa and clover. Again, the rhizobium strain in the alfalfa and clover is specific only to alfalfa and clover. So it's a different strain of rhizobium. As well, we've got the SA, so that means we've also added a strain of trichoderma. Benefits as a crop, or even as a cover crop. The guy's planting a legume, or he's got clover in his cover crop mix. He's wanting to try to produce nitrogen for the next crop. So you increase nitrogen credits, and as a result, with the Graphex SA, we find we get a larger, more fibrous root mass, which helps reduce soil erosion and builds organic matter, which again helps to amplify what he's already trying to do. Here's a study that we conducted up in Wisconsin, and you can see here we have 1, 2, 3, 4-year-old seed, up to 4-year-old seed, and all the seed was already inoculated when the seed was packaged. So what we did here in 2012 was the seed was inoculated that year, but then we also took that same inoculated seed and we overtreated it with fresh ABM Graphex SA for alfalfa and clover. And so what we found is on the first cutting we found a 102% increase in tonnage, or pounds per acre, and then the second cutting we basically broke even. But then when we started looking at 2-year-old seed that was pre-treated 2 years ago, and then we took the same seed that was pre-treated, inoculated 2 years old, and we overtreated it with fresh ABM product, we find at the first cutting we had a 3% increase in pounds per acre, and the second cutting we found we got 5% increase. And so you can go on and see year 4 we had a 9% increase and a 7% increase, and 4 years old seed we found a 15% and a 17% increase in tonnage by overtreating with fresh ABM product. like Graphex SA for alfalfa and clover. So again, how it's packaged, it's a planter box treatment, it's talc graphite. You're going to add 4 ounces per 100 pounds of seed. A case will contain 12 4-ounce packets, and each case then will treat 24 units of seed. And because this is just straight a rhizobium strain that's specific for alfalfa and clover, we can actually get a 2-season dating on the pouch. So if you don't use it, you can keep it in a cool, dry location to keep the shelf life fresh. Which also reminds me that anytime you guys are dealing with biologicals, the biologicals, their biggest nemesis is sun and heat. And so the worst thing you could do is take any like Graphex or Sabrex and put it in the cab of your truck and go out to the field and plant and leave that product sit in the cab of the truck with the sun beating down on the windshield and just baking. What that's doing is just baking the rhizobium and the trichoderma in those packets. So ideally it's better to keep them cool. So ideally if you had a big cooler that you can put in the bed of the truck and put your inoculants in it to keep them cool, that'd be the ideal situation. So you never want to leave it out in the sun and high temperatures and heat, because you're just going to kill the bacteria in it and the fungi in it before you even get it on the seed. We'll move on now to Graphex SA for cover crops. We've got a cover crops product, and it actually is kind of a catch-all for cover crops. We cover everything that a grower might be using as a cover crop. So here we've got peas, lentils, and vetches, so it treats various peas, various lentils, and various vetches. That's the rhizobium strain that's specific for those types of crops or legumes. We've also got the species that's required for cowpeas, lespedeza, and various others that fall into this category. They also have the rhizobium strain that's necessary for true clovers. We've also added on here a trees, all grasses grown for green forage, because we've got the trichoderma. The SA means we've added a strain of trichoderma in it as well. But I would suggest and encourage you to find out all the cover crops that this product treats, is to go to our website and search out Graphex SA, and you can pull the label up and see all the various types of crops that it does treat. And again, when we talk about the benefits of a cover crop, it increases nitrogen credits by producing nitrogen for the next crop. We find we get a larger, more fibrous root mass because of the trichoderma. And again, if there's a grass in the mix, we'll find that you're using grass to help scavenge the nitrogen to save it for next year. So basically, when you're using Graphex SA, you're just going to amplify the effects that you're already trying to accomplish by planting a cover crop. Graphex SA for cover crop comes in 2 package size. Again, it's a talc graphite formulation, and it comes in 4-ounce packets. And that 4-ounce packet will treat 100 pounds of seed. There's 12 packets in a case, and so a case will treat 24 50-pound units of seed. The other container size we have, it's a larger size. It comes in a 40-ounce pail, and that so there's 6 40-ounce pails in a case, and a case will treat 120 50-pound units of seed. We're right at 6,000 pounds of seed. The shelf life on this, again, is only 1 season dating, because there's strains of rhizobium there that we can't get them to live long enough. So we've only got a 1 season dating on Graphex SA for cover crops. That basically wraps up what I had here this evening. Again, I'm the regional rep that covers Indiana, Ohio, and Michigan, so that's why I work specifically with Center Seeds and assisting them and their growers, their dealers on ABM products. If you guys have any questions, you can feel free to give me a call, or you can shoot me an email. And I would encourage everyone to visit our website. There's a lot more information on our website than what I presented here tonight. Also, be sure to click on the blog tab, because in there you'll find there's several articles that have been written about patents that's been pending and the progress and where ABM is headed. And also there's some frequently asked questions too on various products that you might come across when you're out calling growers. So that would conclude.
Hey, Bruce.
Yeah.
This is Eric Belcher. Can you real quick touch on We have some southern farmers in the area that deal with cotton and peanuts.
Yeah.
And I know you have a couple of those products. Can you at least touch base on those so that they know they're out there, and we can definitely get them information on that too?
Yeah, I've got a couple of presentations that probably will take up quite a bit more time than what we were allotted here tonight. But yeah, we do have two products. One is called Graphex SA for peanuts, and again, it's a talc graphite formulation that the grower could add. It's got the inoculant specific for peanuts, plus the SA means we've also added the biological fungi called trichoderma. And then the other product would be Graphex SA for cotton. And again, we've got the legume or the rhizobium necessary for the cotton, as well as the trichoderma in it as well. And I've got two other whole presentations on that if somebody's interested, they can email me or give me a call, and I can walk them through that, and I can even share those presentations with them, along with the yield data that we've seen.
That's great. Well, thanks, Bruce. From now, we're going to...
And Bruce, I got a couple more questions, a couple more things.
Yeah.
Bruce, I got a couple more things I really want you to clarify. You mentioned in there on the graphics for cover crops. In the past, most of the clovers weren't in there. Is that changing, or is that still, the vast majority of clovers that we handle in cover crops, such as your crimson clover and other things, we've typically had to add, had to go to the alfalfa clover mix to get those covered. Is that still true, or?
Yeah, actually, you bring up a good question there. This last winter, we at ABM, we did change the formulation from what this used to be. Instead of what used to be, instead of true clovers, it was actually dry beans. And we found nobody was actually using these small pouches to treat dry beans. So we said, well, it makes sense to just change that over to true clovers, because when you guys were mixing a blend of seed together, you were never putting dry beans, I don't believe, but you're probably putting more of a true clover in there. So we changed the inoculant in there to cover true clovers in there. And clovers, it's the same inoculant for clovers as it would be for alfalfa as well. But yeah, we did change that, and we also added that it also treats all grasses grown for grain and forage.
And then also, Bruce, can you touch a little bit on some of these mixes? A lot of these guys, as they're blending, and they have a 5- or 6-way blend, and you might only have in that blend 2 or 3 clovers, or 2 or 3, maybe a pea, a clover, and a vetch, or something like that. What's your thought as far as the rate to cover everything?
Right. No matter what your mix would be, you'd still want to treat 4 ounces per 100 pounds at the grower level. So if a farmer's out there mixing a seed and treating a seed, he wants to use that 4 ounces per 100 pounds. And ideally, once you treat the seeds, we like to see you get that seed planted within 48 to 72 hours out there at the field. Did that answer your question there?
Yeah, and that's good. And part of the other point there too that not everybody always understands is because it's got the root enhancement for your grasses, it is also benefiting those grasses as well. So you're not necessarily losing if say 50% of your 2,000 pounds is grasses, you're not losing that 50% of the Graphex for cover crops that you're putting on there, because it is benefiting all those grasses. So yeah. Just want to make that point. And then also, Bruce, I had one question back on the corn. You're talking about the yield differences, and you say is some of those did not come back with a positive yield increase. And I know some of Jeff's experience has been that way. Do you have any idea why that might be, or what's causing maybe the instances when you're not getting positive yield increases, I guess, especially on the corn? I mean, we've seen a lot of good results on soybeans, but
yeah, I can give you some idea of what I think might be going on. Sometimes when you do trials, there may be some kind of anomaly that you don't know about until you actually see the data, that something for some reason will cause the yield drop. Sometimes we'll see no yield response, but from what I've been seeing is that SaberX for corn, I think we see a larger yield response when we apply SaberX treated corn in fields that are lower in organic matter, lower in CEC, in fields where it's been continuous corn. I had a guy this year call me up and wanted to know why his SaberX for corn was in this case it was running. He told me 60 bushel more than the untreated, and he also said the corn it's been 25 years of corn, and so that led me to think, okay, this guy's been 25 years of corn, so he's probably got had better germination, better seedling vigor. Plus, we found healthier plants to get that big of a yield response. So basically, to get the biggest yield responses, I see that with low organic matter soils, low CE soils, and continuous corns. I see it on lower productive type ground, basically.
Hey, Bruce, just a quick question for you.
You there?
Yeah, can you hear me now?
Yeah.
Just a quick question on the cover crop inoculant. It said it had a 1-season shelf life. Guys down here in the South, we use our cover crops, and then we'll use that as that same inoculant in the springtime for our grazing covers for cattle. Will that carry over the wintertime, or is a season considered 3 or 6 months or a year?
Yeah, you always want to check. All these packets are going to be stamped on them somewhere, probably on the back side, is the expiration date. So you want to use it before that expiration date. And if you do carry it and it's within the expiration date, you always want to make sure that you keep it in a cool, dry location. And so, depending on ideal location, if you've got spaces, if you've got a refrigerator out in the barn, just stick it in the refrigerator. That's the ideal place for it to go. Otherwise, just keep it cool and dry. Don't let it, just don't let that get to that sun. Don't carry it around your truck for 2 or 3 months. You'll kill the stuff in it when it gets too hot. So keep them dry, and always check for expiration dates on your biologicals and inoculants that you're using, no matter whether it's our product or anybody else's. Does that answer your question?
All right, thank you.
Huh?
Yeah, thank you.
Okay, well, Russell, if you're ready, Bruce, I'm going to go ahead and send over to Russell, and he's going to do he has some photos, and going to do a little bit about what he's doing in the Carolinas and talk about on both sides of cover crop and cash crop.
Okay.
Marty, is the picture up?
Give me a sec.
How's that?
That might share your screen, and then we'll be going. There you go.
I did.
But
You
you
got
look on there. Russell, if you do it one more time, share your screen.
I'm waiting on the icon to come up.
Okay.
How's that?
Yeah, keep going.
Good now?
I can see you,
There
Russell,
we go.
so I think you're good to
Okay.
go.
All right.
Okay.
So what we did, all right. So what we did on our farm is we wanted to conduct some trials using the Graphics SA and some other inoculants that are known pretty much throughout the United States. We did 3 trials. We did the Graphics SA. We used a Root-tastic product, which is a peat, and then we also used a Optimize, which is a seed-coated inoculant. And over these trials, everything was planted on 20-inch rows, and the population was 145,000. We decided at the second and third trifolia we wanted to pull some samples, one to check root growth and plant development, and also early nodulation. The first picture you're seeing on your screen here is the optimize. We'll just kind of scan through a few pictures here. You can kind of see on the next slide here that even with a seed-coated inoculant, we really didn't get that great of nodulation. There's a few on the main stem, and Bruce had kind of alluded to that earlier, that the university trials that we've looked at is nodules on the stem of a soybean plant are 50 to 100 times greater than nodules on a lateral root system. So what we decided on our farm is anything we use for inoculants, we always put on the seed, either in a dry powder or a seed coating, for the reasons that the rhizobia bacteria are mostly immobile in the soil system. So if they're not in that seed trench, you're not getting your maximum benefit. The next one that we'll go to was the Root-tastic. As you can see on the picture, still, even though we applied it on the seed, we really didn't get that great of nodulation. On the next sample, this is our ABM product. You can see the size of the nodules. As Bruce said earlier, they have a really white color, and a majority of them are actually on the main stem of the plant, so we're getting the maximum effect from them. The next slides that we'll show are a comparison side by side. You can see that the plant on the left was treated with Graphics SA, and the one on the right was non-treated. We had a really good fine root system, and you can also see that the taproot was starting to explore more of the subsoil. This is our check plot. Our check plot was the Graphics SA versus a uninoculated seed. This picture was taken at a flower set on our soybean plants. These plants were taken about 4 feet apart, and we had that big of a difference. This is a general picture of how good our field looked with the ABM products. Whenever we did tissue samples, we really weren't lacking in anything on these plants. And a lot of people said that, you know, maybe we just threw out too much nitrogen, and maybe that was the concern. But with the amount of nodulation that you see in these pictures, I don't think that we over-applied. If we would have over-applied, they would have been pretty lazy and wouldn't have set all these nodules. I just think we had such a large nodule population that we really had some good yields out of these plants. And I tell guys, if you're going to use these products, it's best when you use them to go out there and just check and see how this is doing on your farm. Our last picture is going to be our Sabrex for corn. We didn't really get a huge yield increase like some guys have had, but we did have an increase. As you can notice on the left, the plant health was a lot less than on the right. Our check was on the left, and our treated with Sabrex was on the right. The plants tend to stay a little bit longer. If you happen to get in a drought condition, you can usually pick up some later rains and try to put that on for yield. And that's it. Anybody have any questions?
Hello, is anybody there?
Yeah, I'm still
Okay.
here. Bruce.
Hey, guys, if you'd like to go ahead and I could unmute anybody who would like to ask a question, just raise your hand and we'll go ahead and turn that off. Bruce, are you there?
Yeah, I'm still here.
Bruce, go right ahead and ask that question you had earlier. Bruce Cohen. How about Dan Ritter? Dan, do you have a question?
Yes.
Go right ahead, Dan.
Do seed fungicide treatments have any effect on seed treatment for the SA?
The answer is, when you're using Graphex SA on soy seed that's already treated with insecticides and fungicides, there's no issues with that. I have no issues with seed companies and seed treaters that are mixing or high concentrate products with other insecticides, fungicides, and how long it takes them to put it on. Graphex SA, I don't have any problem with any type of seed treatment that's in the market.
Okay. Next I have Nick Voss from Kansas. Nick, are you there? Nick?
Yeah, I'm here.
Go right ahead.
Yeah, the question I have is, we usually double inoculate, even if the seed comes inoculated, we double inoculate. Is there any purpose to double inoculate, like with conventional inoculants, to gain, end production on the soybeans, or is it not necessary with the biologicals?
Typically, we want to double inoculate when we're planting soybeans and field not seen a soybean plant in it for 2 or 3 years. Double inoculate soils that holds a lot of water. So these types of situations, and mostly it's on soybeans going into a field that's not had soybeans in it for 2 or 3 years or more. That's where you'd want to double inoculate. So yeah, you could have your seed supplier inoculate the seed. Then you can add the Graphex SA on top of that to also give you the biological. So you'll have multiple different types of strains of Rhizobium plus Trichoderma with the Graphex SA. If all you're using is Graphex SA and your soybeans that you purchase from your seed suppliers is not inoculated, ABM's recommendation would be to use America's Best inoculant. It's a sterile peat. We want to use a sterile peat to treat the beans, and then to double up, then you use the Graphex SA. You want to recommend the sterile peat just because of the environment that the Rhizobium is living in, and when we get it in the ground, that sterile peat is going to help feed those Rhizobium as well. So ABM would recommend using sterile peat and Graphex SA if your soybeans are not already inoculated.
Okay. Next we have Bruce Cohen from Kansas. Bruce, are you there? Bruce?
Yeah, I'm here. Can you hear me?
Yes, go right ahead.
Yes. Yeah, I've got a question. I've got some family that's using some biologicals on their fertilizers. Wanted to know if using this along with that, if there'd be any issues with that at all.
Okay. Well, the short answer would be no. My other question, though, is I've had, over the years I've been working with ABM, I've had several growers call me and ask that same question. They're using some kind of biological and want to know if our products will go along with it. And I will ask the question, well, what kind of biological is it? What's the active ingredient? What's the living organism? What's the concentration of it? And multiple times I get a response that, well, I don't know what it is. And so they'll tell me what it is, and I'll Google it and look at the label, and I've come to find out it doesn't have any biological whatsoever in it at all. No living organisms, no microbials in it. But basically what it was a lot of times it's just like it's a micronutrient that feeds living or microorganisms. So I've never heard of anybody mixing a true biological with a fertilizer and applying it that way. but there may be something that they're adding to the fertilizer that's that's a food source for microbial activity in the soil. So for ABM, though, we like putting ours on the seed. All of our products are seed applied, so we get a concentrated level of bacteria and fungi right on the seed where it needs it the most, so it doesn't have to go out and look for it in the soil profile. So you need to ask questions sometimes to get down to what is it they're trying out, and what's the active living organism in that product, and what's the concentration level. So that's what I would do if I was talking to that grower. I'd want to know what exactly they're using.
Okay.
Do you have a follow-up, Bruce? No.
No, that's
Okay.
great. I just was gonna. I would like him to maybe contact me, or I'll call him or something one of these days and discuss that.
Okay.
Sure.
Hey, next up is question is from Russell over in the Carolinas. Go ahead, Russell.
Hey, Bruce, I got a question. Our university here said that after 3 years of soybeans that I didn't need inoculants. After talking to other universities, I've heard that if the soil gets above 90 degrees or below 30, if we get a sustained drought, or if we even get flooding and the soil is actually saturated or underwater for more than a couple days, that those rhizobia bacteria can actually take a hit, so to say, and you're actually going to lose some of those colonies. So it's good to pretty much re-inoculate every year or every other year, depending on your rotation. Have you heard anything like that, or seen any university data?
Yeah, and if you want to switch me back to a live screen here, I've got some other slides I can share.
Hold on a
that
second.
would relate to that. Yeah, when you keep in mind that these microorganisms are sensitive, and so when we they're sensitive to extreme heat, they're sensitive to drought situations. They're sensitive to moisture. If you get too much moisture in the soil profile, you can kill off. But what you're talking about is what Dr. Jim Berline of Ohio State University. He worked at Ohio State for many years, and he specialized in soybean inoculum and studied soybean inoculation for quite a while. And can you see my screen now? Is my screen up on everybody?
Yep, we're up. Mm-hmm.
Okay,
Yeah, I can.
So here's some stuff that Dr. Berline has shared with us out here up in Indiana and Ohio. So here's some studies that he's conducted, and basically what he says is that, not every year are you going to see a statistical yield advantage by inoculating your soybeans. And here you can kind of see this year 2001, 2, 3, 4, 5, and 6. And year 2001, there was 14 treatment numbers or replications, and the UTC would be the untreated check. Then you would have the inoculated yield. And you can see right here in 2002, there was no yield response at all in 2002. In 2003, there was only a bushel and a half. But when you take all these years and all those replications and average them out, he found that's a 3 bushel yield response by inoculating your beans over a period of time. And the analogy that I can put with this, it's kind of like investing in a 401k program or an IRA. You put money in it every year, but not every year do you see a gain in your investment. But over a course of time, you should see a gain in response. We talk about ideal the soil conditions. So here's some things that he's talked about that they've learned over the years. Well, ideal soil with ideal soil conditions, we can find pink nodules 21 to 25 days after emergence. And inoculation can increase yield by 12 to 15%, with certain field conditions being 5 to 8%, which is more typical. Here again, he says inoculation may not always produce a statistically significant yield increase, but it will be very profitable over time. And again, soil texture, tillage, and organic matter have little effect on the productivity of soybean inoculation. And the more ideal the soil conditions for rapid seed germination, the more effective inoculation will be. And then to add to this too, a lot of times when you say there's 3 years of the field having soybeans in that field, yeah, you're going to build the rhizobium population up. But what Dr. Bierlein calls, that's called resident rhizobium. That's the rhizobium that hangs around in the soil, okay? And it's kind of like resident geese. You know, here in Indiana, we see geese flying from the north down south, but we got some geese that just want to stay around here year-round. We call that resident, and they're lazy here too. And what he's found is they produce less nitrogen than the improved cells do, the improved rhizobium cells. And then some of these rhizobium then produce no nitrogen at all, even though they receive nourishment from the plant. So some they're actually robbing from the plant. Some other things that might that would affect nodulation, and I got another chart here. It shows here, this is a soil pH effect on nitrogen fixation. So when we planted the soils low in pH, this study here, they found 0 nodules were produced by the plant. And then when we get up above an 8.2 soil pH, our population of nodules on the plant starts to decline. So on the high end and the low end of the soil pH are the 2 toxic sides that affects nitrogen fixation, or the nodulization on the soybeans. So ideally, we want between 6.5 and 7.4 for soil pH. Another thing that's going to affect nodulation, at least early on nodulation, is the nitrate carryover in the soil. So if you find, I know there's some farmers out there that like to go out and put a little bit of nitrogen on their soybean fields. And from what I've learned, I think that they're probably delaying the onset of nodules on the soybean plant, because when a soybean plant is able to take in nitrogen, a little bit of nitrogen that is in the soil, then it's thinking, well, why do I need to put nodules on? And so it delays putting nodules on. And so in my mind, I'd like to see nodules get on those soybean plants just as fast as possible. So even though one of our strains likes the soils that might have nitrate carryover, you got to keep in mind that that's 1 strain out of 3 in that scoop that you're adding to that soybean. So even then, you've only got 1 that's really taking advantage of that nitrate carryover. So those are some things that I've learned from Dr. Bierlein, and like he says, over time, you may not always see a yield response, but over time, it'll pay off for you. Does that answer your questions?
Yeah, thank you.
Is there anybody else who has any question? If you want, just raise your hand. I'll unmute you. Anybody? Okay, well, if there's no more questions, Bruce, I'd like to thank you for stopping in, taking time out to talk to us. If you want to put your contact screen up, so maybe whoever's online here can take a peek at it and write your number down, and if they want to give a call or drop you an email, they could do that.
Yeah, can you see her there?
Not yet.
There you go.
There you go. All right, give that a minute there.
Yeah.
There's his email and phone number. As always, you contact through him or through our regional reps. And once again, thanks, Bruce. We appreciate it. And we look forward to talking to you down the road about more ABM.
Hey, Marty, I just got one last thing before everybody gets off. Again, this is Eric Belcher. This year, what you guys are going to see is you're going to see a whole lot more of these webinars coming up. I do appreciate everybody being on tonight. I know Rob right now is working on a whole host of webinars, and we're planning on getting out a schedule to you fairly quickly here. But what we want to do is be able to provide information like this. It's going to be on a whole host of different things, but at least we're aiming for about 1 a month. Some months we're going to try to have more than 1 a month. So if you guys can make it, great. If not, I know we're going to be recording all these, and we'll have them available for you as well if for some reason you can't make it. But again, I appreciate everybody being on tonight, and hope you guys took something away from tonight, and and we'll be able to improve your guys' farms specifically, and then also your customers' farms as well. So, again, I thank you.
Thank you.
All right,
Appreciate it, guys.
Thanks for having me.
Yep.
Thanks.
I'll take care.